Hi, thanks for joining us. I'm Casper Mossman. I'm the communications director at QB3 and Baker Labs. And Baker Labs, we're doing our webinar today live from Baker Labs. And here behind me, you can see many of the tenants from our UC Berkeley incubator. Thanks. And our tenants say this is a bioscience incubator based at UC Berkeley in the former art museum. We've got tenants here developing new technologies in areas such as pharma therapeutics, research tools, biologics, new foods, gene therapy. And we currently have about 27 companies in-house. We have a capacity for 35 to 50, depending on the size of the company. And a distinguishing feature is in keeping with the Berkeley spirit, Baker Labs has a double bottom line, which if you haven't heard of this concept, it means that they're for-profit companies, but we select our tenants based on whether they're doing something which could benefit society. So, you know, no gaming apps here. I'd say we were excited, but, you know, excited doesn't really begin to describe our excitement at having Dr. Carolyn Bertozzi here with us. Carolyn Bertozzi is the Ann T. and Robert M. Bass Professor of Humanities and Sciences at the Baker and the Baker Family Director, that's B-A-K-E-R, Family Director of Seraphin Chem-H at Stanford University. She's also an HHMI investigator. Her research focuses on profiling changes in cell surface glycosylation, something that scares off a lot of scientists, including myself, associated with cancer, inflammation, and infection, exploiting this information to develop diagnostic and therapeutic approaches, most recently in the areas of immuno-oncology. Dr. Bertozzi earned her PhD at UC Berkeley and did a postdoc at UCSF. This is very important to us because I work at QB3, which is UC Institute. Took a faculty position at Berkeley for moving to Stanford in 2015. She's an elected member of the National Academy of Medicine, National Academy of Sciences, American Academy of Arts and Sciences, also been awarded the Lemelson MIT Prize, MacArthur Foundation Fellowship, Chemistry for the Future Solvay Prize, and finally, we've all been waiting for it, with Morton Meldal and Barry Sharpless, the 2022 Nobel Prize in Chemistry. Congratulations, Dr. Bertozzi, and welcome.
Thanks, Casper. I know there's an echo there.
I'm turning it off.
Okay, great. So first of all, I really appreciate the invitation to come and present in this webinar series. We set this up a long time ago, and the timing turned out to be interesting. And I was worried 2 weeks ago that my calendar would get so upside down that I wouldn't be able to fulfill my commitments like this one. But I worked really hard to guard this time slot because I wouldn't want to miss this for the world. Mostly because, you know, the work that I was recognized for 2 weeks ago was work that was entirely done at UC Berkeley, where I was on the faculty from 1996 all the way to 2015. So that's almost 20 years. And I thought it would be fun to share the story of bioorthogonal chemistry as it began at UC Berkeley, and then to transition in the second part of the talk to some more translational work that we've been doing, because it kind of fits with the spirit of QB3 and the new Baker Labs. So with that, let me share my screen. So what I'm going to talk about is the basically the origin story of bioorthogonal chemistry, why we decided to go on the journey to develop these kinds of reactions in the first place. And as you'll see, it has to do with a longstanding interest that I have had in the field of glycoscience. And then at the end of the talk, I'll show you a new cancer immune therapy candidate that we developed, which was enabled by bioorthogonal chemistry and which targets changes in the glycocalyx, which is the cell surface sugars that accompany cancer. So let me just start by saying that as a chemical biologist, I'm always looking for problems in biology that might be uniquely solved with chemical approaches. And my experience in that area is that sometimes the biological problem at hand can't be solved with any current technologies, and that motivates the chemist to try and invent new technologies. And this interface between technology development and biological problem solving is one that I love and many chemical biologists really enjoy. So for us, the technology had to do with developing chemistries that you could perform in living systems. And what I mean by living systems are environments as complicated as a living cell, or a laboratory animal, or even a human patient. And to do chemistry in settings like this was something that really had no precedent back when I joined the Berkeley faculty in the mid-1990s. At that time, you could pretty much describe the field of organic chemistry through, for example, Richard LaRocque's Comprehensive Organic Transformations book. This is a book that populated the shelf of many of us organic chemists at that time. And it basically lists thousands of organic transformations, every single one of which was developed with the assumption that you could perform the chemistry in an environment where you have precise control over every parameter. And that's exactly what happens when you do chemistry in the conventional vessel, like, for example, this round bottom flask. By contrast, if your goal is to do the chemical reaction in a living system, you have to relinquish all of the control that the laboratory and the fume hood and the round bottom flask give you. And you have to develop chemistries that have such high degrees of selectivity and biocompatibility that they won't be offended by all of the complex biological molecules in these more complex systems. So that motivation is what drove the development of what we call bioorthogonal chemistry. And you can define that as chemistry that does not interact or interfere with a biological system. And as I'll show you, in thinking about how to develop these bioorthogonal reactions, the first step is to take inventory of all of the molecules that define biological reactivity space. And there's a lot of functionality in biology, to be sure, but it's not infinite. There is a boundary around it. And just as there might be a galaxy of biological reactivity space, we wondered whether there could be a separate galaxy with other functionalities that don't exist in our natural world. And maybe those functional groups could create the bioorthogonal reactivity space with lots of interesting applications that would ensue. So the problem that drove us to think about bioorthogonal chemistry had to do with glycoscience. And even at the time that I started my job at Berkeley, already there were at least 30 years of literature from oncology showing changes in cell surface glycosylation that are characteristic of cancer cells in the tumor microenvironment. And in fact, that literature was so deep that many people at that time wondered whether changes in cancer cell surface glycans could be used as diagnostic markers. And then even more deep, what the functional significance of those changes might be, and the possibility of discovering new therapeutic targets. But at that time, all we knew is that in many different tumor settings, what is normally what I think of as a normal pattern of glycans that terminate with a particular sugar called sialic acid, and that's the pink structure here, that pattern shifts and you end up with an overproduction of sialoglycans on different kinds of glycoproteins and glycolipids. But this change in the abundance of cell surface sialic acids was so prominent in so many different tumor settings, the chemist couldn't help but wonder whether that molecular difference between the disease and the healthy tissue could be exploited for diagnostics or therapeutics. So at that time, we thought molecular imaging might be an area where one could capitalize on changes in cell surface glycosylation. And when I first started thinking about this, I was a postdoctoral fellow at UCSF working in Steve Rosen's lab. He was an immunologist. And this is the time when the green fluorescent protein had really hit stride. And Roger Chen's work at UC San Diego was recognized with a Nobel Prize. And beautiful technologies like the green fluorescent protein and various other imaging modalities that focus on nucleic acids, for example, ions and metabolites. These kinds of imaging platforms were really transforming our understanding of biology because you could use them to study biological molecules in living systems, in live cells, in model organisms. And at that time, there was really no technology to image the cell surface sugars. And we thought, given how they change in cancer, if you could image those sugars non-invasively in living systems, you might have a new way of detecting cancer. So we came up with this chemical strategy, which is illustrated on this slide. And the idea was to leverage metabolism to incorporate unique chemistry into cell surface sugars. And we were focusing on sialic acid because of its cancer relevance. And so we thought maybe an unnatural synthetic sugar that is a metabolic precursor of sialic acid could be chemically modified with a functional group. And so here, let's just call it X. And if we could find an X that was inert in the biological system, small enough so that metabolic enzymes might not notice it, and also that it wouldn't perturb the natural functions of these sugars, then maybe that X group, after a metabolic process, would end up in cell surface glycans, where it is now available for chemistry with an imaging probe. And that imaging probe would have its own complementary functional group Y, where X and Y would react to form a covalent bond. And now that probe would be a proxy for the sugar of interest, or the sialic acid. So that's simple on its face, but of course the big challenge with this concept was identifying the functional groups X and Y, which could do chemistry with each other in a living system. And that was the motivation for coining this term bioorthogonal, and then for looking for the right chemistry to execute the imaging experiment. So, we spent time thinking about all of the complex chemistry of a cell and looking beyond all of that to other kinds of functionalities which don't have any natural counterparts. And in the early days, we focused on this functional group in the middle of the crystal ball here, which is an azide, and the various chemistries that azides can undergo. And I'm going to describe them in more detail, but we focused on alkyne cycloadditions. But over the years, lots of other functional groups have now emerged as being useful for in vivo bioorthogonal chemistry. And if you want sort of a broader view of the field, I would encourage you to read Neil Devarage's review article, from which I borrowed this image, which was published in ACS Central Science a few years ago. But starting with the azide, when I was a grad student, I was a synthetic chemist, and I built amino sugars for various applications. And the standard approach towards synthesis of amino sugars was to use azides as a kind of protected version of an amine during the synthesis. And I had used azides in this context, and so I knew that there were some really selective and mild ways of reducing azides to amines. And one of those was the classic Staudinger reaction with triphenylphosphine. So if you take an alkyl azide and react it with triphenylphosphine, those two will join to form an intermediate called an aza-ylide. And the driving force for that reaction is the extrusion of nitrogen gas. You can do these reductions in aqueous buffers. And when you do that, this aza-ylide will hydrolyze spontaneously to give usually the desired product, which is your primary amine, and the oxidation product, triphenylphosphine oxide. And what I loved about this reaction is that the azide is a soft electrophile, the phosphine is a soft nucleophile, and neither of these functionalities will react, at least at any reasonable rate, with natural biological functional groups. They react very selectively with each other, and they'll do this in water. So this very first step of the Staudinger reaction, in a way, looked like a bioorthogonal chemistry, where the two reagents react to form a covalent adduct. But the problem is it's not stable in water. So we felt if we could find a way to make aza-ylides stable, or react in a way that keeps them together in a stable linkage, then we would have a useful bioorthogonal chemistry for execution in living systems. And one of my Berkeley students from the early days when I was still an assistant professor, Eliana Saxon, she's the one who pulled this off with simply adding this methyl ester ortho to the phosphine on one of the aryl rings. And so now when this type of phosphine reacts with an azide, the aza-ylide is poised for an intramolecular reaction, a cyclization reaction, where the nitrogen attacks the carbonyl of the ester, liberates methanol as a leaving group to generate this amide-linked intermediate. So now when the phosphorus nitrogen bond hydrolyzes, there's no crisis because the amide keeps the ligation product together. So we called this the Staudinger ligation, and it was the very first example of a bioorthogonal chemistry. And we were able to use this chemistry to monitor sialic acids in living systems, first in cultured cells, but eventually in mice. And that was the beautiful work of former grad student Jen Prescher, who's a professor at UC Irvine now and very well known for her own contributions to bioorthogonal chemistry, as well as Danielle Dubey, who's now a professor at Bowdoin College, where she also continues to do glycoscience with azide sugars. But in this particular study, what these two chemists did is they prepared this azidoacetyl derivative of a known sugar called N-acetyl minosamine. So the natural sugar is called MAN-NAC. We abbreviate this sugar MAN-NAS. And by peracetylating this sugar, it's lipophilic enough so that after you inject it into animals, it will basically enter cells by diffusion across plasma membranes. And then the acetyl groups get cleaved intracellularly to liberate the free sugar. And there's a roughly 6-step biosynthetic process that converts MAN-NAS to the corresponding azidoacetyl sialic acid, or SiaNAS, on cell surface glycans. And the azide just comes along for the ride and does no harm, and it's just waiting there for the moment that it can encounter its reactive partner. And so, for example, triphenylphosphine derivatives with these methyl esters conjugated to probes that allowed us to detect these adducts in vivo turned out to be useful for profiling sialic acids on cell surfaces. And this was all a very exciting time in our lab. However, there were some issues with this chemistry. Most importantly, although it was really selective and very bio-friendly, no toxicity or anything like that, it was kind of sluggish. It had somewhat slow kinetics. And as a consequence, we really couldn't use the chemistry to image changes in glycosylation that were happening on fast timescales. So while we were using the Staudinger ligation for various applications, we also understood that a faster bioorthogonal chemistry would have its own place and could help us do different kinds of experiments. So that's when we started thinking about other manifolds of reactivity of azides. And, you know, another great thing that azides can do is 1,3-dipolar cycloadditions with alkynes. And when they react in that way, they'll generate a triazole product. And this was chemistry that was spearheaded by Rolf Wiesken, a German chemist who just passed away, about 4 years ago before COVID. He lived almost to the age of 100, and I had the great fortune of meeting him about 10 years ago at his home institution, which is University of Munich. So Rolf did a lot of mechanistic work on this reaction in the previous century. And one thing we learned from reading his publications is that there's a pretty high kinetic barrier to the cycloaddition reaction, even higher than the Staudinger ligation. So this chemistry is also very slow at room temperature or physiological temperature. And when people do this chemistry in the lab, they tend to have to boil the reaction like above 100 degrees C. So that's why it was such a big deal when my Nobel Prize partners, Barry Sharpless and Morten Meldal, published 2 landmark papers almost within just a few months of each other back in 2002. And what they had shown here is that you could catalyze the cycloaddition of azides and terminal alkynes with a copper catalyst. And this so-called copper azide alkyne cycloaddition, which is now almost used synonymously with the term click chemistry, this reaction does go very fast at biologically compatible temperatures to form the corresponding triazoles. And in the 10 years since these 2 papers were published, this copper catalyzed click chemistry has become probably one of the most important reactions in chemical biology. But as exciting as that was, it really was not on trajectory to help us solve our problem. And that's because the copper catalyst that's required for this is cytotoxic. So this is not a chemistry you would want to perform on live cells or could probably ever perform in living organisms. It wasn't going to solve our problem. So the real breakthrough for us came when we realized that there were other ways to accelerate the azide alkyne cycloaddition that wouldn't require a metal catalyst. And the paper that really pointed us in the right direction was this wonderful study from 1961 from Wittig and Krebs, more German chemists from the previous century and also very famous names. And they were studying dipolar cycloadditions with phenylazide. And one of the substrates they tested was this bizarre looking alkyne in a ring system. So this is cycloalkyne. And unlike regular alkynes that are not in a cyclic system, which react very slowly, this alkyne reacted with phenylazide, as this German line would suggest, which is like an explosion, okay? So we realized that the strain energy associated with putting an alkyne in an 8-membered ring, which contorts the bond angles away from linearity, and it's about 19 kcals per mole of strain in a ring like this, that strain energy would make this alkyne a lot more reactive. And perhaps this was the type of alkyne you could react with an azide at physiological temperatures. So I had a series of former Berkeley students and postdocs, Nick Agard, Jeremy Baskin, Scott Laughlin, Ellen Slatton, John Jewett. All of them now have positions at these various institutions, but they sort of worked in tandem and collaboratively with each other to build a series of variously substituted cyclooctines in order to tune the reaction kinetics with azides so that they would be ideal for our imaging applications. And there's a lot of structures on this slide. The numbers below each structure are the second order rate constants for the reaction with benzyl azide. And I just highlight a few of these structures to illustrate the points. If you have a relatively unfunctionalized cyclooctine, the second order rate constant is in the 10 to the -3 per molar per second range, which is kind of on par with the Staudinger ligation second order rate constant. But you can boost the rate significantly by putting electronegative substituents next to the alkyne, as illustrated by this reagent called DIFO. There's about 2 orders of magnitude enhancement in the rate constant. You get another boost if you increase the strain a bit by fusing benzene rings to either side of the cyclooctine. And now we're about 3 orders of magnitude up from the basis set. And then finally, if you really want to get fancy, you can shrink the ring down to a 7-membered ring. You can't do that with an all carbon ring system because it's just not stable at room temp. But if you relax the strain by putting a larger atom in the ring, like a sulfur atom, and then put gem dimethyl groups flanking the alkyne, you have a reagent that is a very fast dipolar cycloaddition substrate with azides. So now we finally had a chemistry that could basically leverage what we already had done with the azide, but allow much faster bioorthogonal reactivity in living settings. And by the way, I should point out that there was a lot of really fun physical organic chemistry that accompanied this work. And there were people like Ken Houck at UCLA, who's very well known for his work in theory and transition state modeling. And he was sort of on the hunt for interesting new reactions to study mechanistically. So when we published on the cyclooctine reactivity with azides compared to linear alkynes, he said, hey, let's do some computational work and try and understand the origins of this beyond the simple explanation of ring strain. So we had a fun time publishing this work together in 2012, understanding how, in his parlance, the transition state distortion and interaction energies contribute to the boost in kinetics of the cyclooctine. Okay. But meanwhile, we finally had a reagent that we thought could perform imaging studies in living animals. And the first thing we did was take this system into a model organism that is very amenable to optical imaging, that being the zebrafish. And many of you probably know that zebrafish are a really wonderful model for developmental biology because you can generate embryos by in vitro fertilization and watch the developmental process from all the way from a single cell to a 5-day-old fish. And all along the way, you can have them under a microscope and you can see how molecules change if you can label them with fluorophores. So with our newfound power of bioorthogonal chemistry, we were able to label various glycans in the zebrafish larvae during the developmental process. And not only did we see some really striking patterns of differential distribution of different types of glycans, which in this image here, there's different glycans with different colors, but even right down to the single cell level, we could actually look at rounds of cell division in the early embryos and watch the glycans on the plasma membrane redistribute themselves during the course of the cell cycle. So that was a really exciting period of time in the lab because we finally were able to do imaging of cell surface sugars, which is the goal that we had set out before the project even started. And then a lot more fell from there. So, the same exact platform was useful to image glycans in other model organisms like C. elegans. We did some tumor imaging studies in mice. We imaged bacterial peptidoglycan, both in vitro and in vivo. Others used the same platform to image cell walls of Arabidopsis and to look at changes in cell wall structure for different kinds of mutants. And then at Stanford, we've done some work where we harvest tumor tissues from patients. And this here is a prostate tumor, freshly excised from a patient at Stanford Hospital. And we can do the metabolic labeling with azido sugars in organ culture, and then tag those sugars with various probes to do biomarker discovery and glycoproteomics. So, all that work is published now. And, if someone wants to go back and kind of review its history, I'm certainly happy to point you to the primary literature. But I thought it might be fun to now shift gears and talk about the work that we've done more recently, and in particular, since I moved my lab to Stanford back in 2015. Because right before that move, we had started a project aimed at understanding why it is that cancer cells undergo this change in their cell surface glycosylation pattern. And again, I started the talk by telling you that normal healthy cells have some amount of sialoglycans on their cell surface, and it's like a well-manicured garden. But it's often observed that in the setting of a tumor, the cancer cells have an unruly jungle of glycans on the cell surface, and they undergo an upregulation of these sialoglycans. And why on earth would something like that happen, was the open question at the time. So back around 2009, 2010, there was a huge resurgence of interest in the field of cancer immunology. And at Berkeley, we know that Jim Allison, our former faculty colleague, was a major player in this space. And in fact, he invented an immune therapy at Berkeley, which ended up being a clinically approved drug, and it's now really changed the standard of care in oncology. So what was learned is that our immune system plays a very important role in protecting us from cancer. And the way that works is when a cell is becoming damaged on the path to becoming a cancer cell, it will present signatures of that damage on the cell surface in the form of different molecules. Immune cells have receptors that allow them to sample those molecules through the formation of a synapse. And in this way, the immune cell can collect information about the cancer cell or the healthy cell, if it is one, and make a decision about whether to leave that cell alone or to get rid of it. And so a simplified view of that synapse is here. And what's happening at the synapse is there's two different types of receptors on the immune cell. Some of those receptors are activating. And what that means is if they cluster at the synapse in complex with their ligands, they'll deliver a positive signal to the immune cell, which tells that immune cell to fire off a response and ultimately kill the target cell in the synapse. And different immune cells have different ways of killing their target cells. So, a killer T cell will do a direct kill, and an NK cell or a natural killer cell, similar, just a direct kill by secreting factors that punch holes in the target. Whereas a macrophage, different type of immune cell, will phagocytose the tumor cell and eat it up and digest it. But in any event, you need to have strong signaling through activating receptors to initiate that. And so the ligands that those receptors are looking for are signatures of bad health, signatures of cell damage. Now, it's kind of risky for an immune cell to have a pathway like this, because what if it makes a mistake and reacts against your healthy cells and starts killing healthy cells? When that happens, you end up with an autoimmune disease, right? So there's a built-in mechanism to try to protect us from autoimmunity, and that's this other type of receptor. And these are the inhibitory receptors. And so immune cells have these receptors that when they engage their ligands on the target they deliver inhibitory signals, which literally quench the activating signals and prevent the immune cell from reacting. And as you might imagine, if a cancer cell, just through the process of natural selection, ends up in a phenotype with high copy numbers of these ligands for inhibitory receptors, well, that's a cancer cell that can survive immune surveillance and live another day. And so it might not surprise you to learn that if a cancer has progressed to the point where you have palpable or visible tumors and disseminated tumors across the body, chances are that cancer has figured out how to escape recognition by the immune system through the expression of ligands for these inhibitory receptors. Now, the inhibitory receptors that are by far the most recognizable nowadays are the so-called T-cell checkpoint receptors. There's two that have been successfully drugged, and they are PD-1 and CTLA-4. And basically, here's a blow-up of the biology, again, in honor of Jim Allison. PD-1 and CTLA-4 have signaling domains in the cytosol of the T cell. And there are motifs within those signaling domains called the ITIM and ITSM motifs. Those are acronyms. The details aren't important, but it suffices to say that they're regions of the protein that dock onto phosphatases called SHP1 and SHP2. And these phosphatases are kind of like the biochemical actors in the quenching of an activating signal that might come through an activating receptor. And on the T cell, the most important activating receptor is the T cell receptor, which is recognizing neoantigenic peptides presented in MHC molecules. And those neoantigenic peptides are basically fragments of mutated proteins, which are a reflection of mutated DNA. So this is the indirect way that a T cell can sense whether there's mutated DNA in a target cell. And that's a cell the T cell wants to kill, because too many mutations in your DNA, that's on the path to cancer. But like I said, through natural selection, if a cancer cell is able to express PD-L1, which is a ligand for PD-1, or B7, which is a ligand for CTLA-4, then it's able to deliver inhibitory signals that quench the activating signal. And that's of benefit to the cancer cell. So from a therapeutic standpoint, this was a really important discovery because it led several groups, inspired by Jim Allison's drug now called Yervoy, to develop monoclonal antibody therapies, where the antibodies bind either the inhibitory receptor, PD-1 or CTLA-4, or the corresponding ligand, such as PD-L1, and block those two proteins from engaging at the synapse. And, Merck is the owner of the drug Keytruda. That is now, probably the number 2 or 3 selling drug in the world. It's become standard of care for many different tumor types. It and Yervoy together were the 2 leading immune therapies, both approved back in around 2012. And since that time, lots of other companies have come in with their own variants of these antibodies, and it's just a major industry. So I took the time to review all this biology because it was in reading this story as it unfolded at that time that gave us an idea for what we thought was potentially another very important system of immune modulatory receptors and targets for immune therapy. And those are the sialic acid binding Ig-like lectins, which is kind of a mouthful. So we abbreviate this family of immune modulatory receptors, the Siglecs. So this is a cartoon that shows the domain architecture of the human Siglecs. There's 14 of them in humans. And if you picture this yellow line being the membrane, the plasma membrane of an immune cell, outside the ectodomain of these receptors comprises a variable number of Ig domains, which are these blue ovals. And then there is a domain that binds sialic acids. So these are sialoglycan-binding receptors, and that's the crescent moon. What really struck us is what you find inside the cell, inside the immune cell, in the cytosolic domain of these proteins. Because many members of this family have the so-called ITIM domain, and the 4 in the middle here, Siglec 7, 8, 9, and 10, have tandem ITIM, ITSM domains. And if you just took the amino acid sequences in these cytosolic domains and did a blast analysis of the human proteome, out would come PD-1, the T cell checkpoint receptor. So it turns out that the Siglecs and PD-1 have basically the same signaling biochemistry. They suppress immune cell function. But whereas PD-1 is pretty much, expressed exclusively on the T cell, these members of the Siglec family are found on virtually every type of immune cell. So, for example, you can find Siglec-7 on natural killer cells, macrophages, and alongside PD-1 on activated T cells. Siglec-9, you find it on various myeloid cells as well as lymphocytes. And for any given immune cell type, there's usually more than one Siglec that's expressed on that cell. So we hypothesized that the Siglecs could be a major axis of immune modulation. And just as PD-1 engaging PD-L1 allows suppression of the T cell, maybe sialoglycans engaging the Siglecs can induce suppression of lots of different immune cells. And if so, this might explain why so many cancers have been found to have upregulated sialoglycans, because that's a phenotype that would be advantageous as it would allow evasion of immune cell killing. So back at Berkeley, around 2010 or so, we started a project to test this hypothesis experimentally with the hope that, when patients don't respond to T cell checkpoint blockade, which most of them don't, maybe some of those patients instead would respond to an immune therapy that blocked engagement of the Siglecs and the sialoglycans. So where we started with our experiments was with NK cells and macrophages, which contribute to anti-cancer immunity through a process called antibody-dependent cell cytotoxicity, which is abbreviated ADCC. And it's well known that when cancers arise in our bodies, our immune system can sense antigens on the surface of those cancer cells as being unnatural or, not healthy. And we mount an immune response against them, which can include the formation of antibodies. So many cancer patients actually have antibodies in their circulation, which are anti-cancer antibodies. And when those antibodies are contributing positively to immunity, one of the mechanisms is to allow synapse formation with these NK cells or macrophages through engagement with the Fc receptor, Fc gamma R3. And so the antibody basically forms, a bridge between the cancer cell, the Fc receptor, and the immune cell. And a synapse of this type is a very activating synapse that would normally lead to killing of the cancer cell. And the question we asked in this early study was, suppose the cancer cells are heavily sialylated. Well, in that setting, in principle, they could recruit a Siglec to the synapse. And for NK cells and macrophages, there's Siglec-7 is a pretty preponderant Siglec. And with its ITIM and ITSM domains, Siglec-7 could in principle quench signaling through Fc gamma R3 and inhibit the activity of that immune cell. And we published this back in 2014 because this is exactly what's at play. And in fact, it's a resistance mechanism for cancer cells against ADCC. And furthermore, not only does this allow cancer cells to evade the endogenous antibody response, but patients who are treated with monoclonal antibody cancer drugs like Herceptin, the anti-HER2 antibody from Genentech and Roche, or Rituxan, the anti-CD20 antibody used to treat leukemias, first developed at Stanford and now a longstanding approved drug. These are antibodies that work for some patients and not for others. And it turns out when these antibodies fail, it's often because the patients have heavily sialylated glycans on the surface of the cell. So after we published that back in 2014, which is this paper up here in the upper left, there was a whole series of publications from other labs looking at different Siglecs and different cancer settings with different ligands, all the way up to 2019, where my own Stanford colleague, Irv Weissman, looked at Siglec-10 binding to a glycoprotein CD24 in ovarian cancer. And there was so much excitement during this 5-year period about this new mechanism of immune suppression, which was being validated, that C&E News did a really nice piece naming Glycoscience as the fuel for a new breed of cancer immune therapies. So by this time, we were pretty confident in the importance of the biology, and we were starting to focus on therapeutic strategies. So, how do you block the interaction between sialoglycans and the Siglecs to potentiate the immune reaction against cancer? And, on the one hand, you could just take a page from the playbook of Keytruda, right? Or the PD-1 blocking antibodies. And, you could develop an antibody against the Siglecs that binds, with a footprint that prevents their engagement with the sialoglycans, right? That's a reasonable approach. But the problem is, there's not just one Siglec, there are many Siglecs. And, once we started looking at their expression patterns, we realized multiple Siglecs could contribute to immune suppression through this axis. And, you might have to block more than one Siglec to get the full immune potentiating effect. And how do you know which one or ones to block for any given particular tumor? I think these are questions that would be really interesting to answer, but at the time we were working on this in the early days, this was a book that hadn't been written yet. So rather than trying to pick a Siglec target or a combination of Siglec targets without really the knowledge you would need to do that intelligently, instead, we decided we would focus on the cancer cell and go after the ligands for the Siglecs. Now, to be fair, there wasn't a whole lot known about the actual biological ligands on tumor cells in the actual setting of the disease. The only thing we knew for sure is that whatever the Siglecs are binding to, whether it's glycoproteins or glycolipids, there had to be a sialic acid residue as part of that ligand. There's a hotspot in the Siglec binding domains that's conserved across the family, and engagement with the sialic acid contributes a lot to the binding energy. So we had this idea that we would develop a therapy that literally removes sialic acids entirely from a cancer cell. In other words, a therapy that degrades cell surface sialoglycans, kind of like a molecular lawnmower. And if we could make a therapy that could cut sialic acids off of cells and target it exquisitely to cancer cells, then in principle, we would deprive those cancer cells of ligands for any member of the Siglec family. So the form that lawnmower took was an antibody-enzyme conjugate using enzymes called sialidases. And this is a well-known class of enzymes. We humans have sialidases. We have 4 of them that we know of encoded in our genomes. They're also found in microbial genomes, and viruses might make them, like the influenza virus. So there was a lot of literature to go on in choosing or engineering the right kind of sialidase for this type of molecule. And so we made a prototype in which we fused 2 copies of a salmonella bacterial sialidase down near the C-terminus of the heavy chain of the approved drug Trastuzumab. It's an antibody. It's the active ingredient in Herceptin. And it's an antibody that binds HER2, a protein overexpressed on breast cancers and gastric cancers and lung cancers, a very well-trodden cancer antigen with an effective antibody therapeutic. But we thought by putting a sialidase on trastuzumab, we could strip sialic acids off of HER2 positive cells and render them more susceptible to killing by the immune system, which would be therapeutically, complementary to other HER2-based therapies like the naked antibody or some antibody drug conjugates that are out there that are based on trastuzumab. And this was the work of former grad student Melissa Gray. She's now a postdoc in Stephen Bannock's lab, my colleague in Stanford Chemistry Department, as well as a former postdoc named Han Shao, who is now doing some really innovative work in next generation antibody drug conjugates in his own lab at Rice University. So now I have an opportunity to show you how our earlier work in bioorthogonal reaction development intersected with this more recent work in cancer immune therapy development, because the way that we attached the sialidase to the antibody in our prototype molecule was through chemical conjugation. And the selectivity and biocompatibility of these bioorthogonal reactions made them perfect for this type of a synthesis project between two proteins. So to get there, we actually used two different bioorthogonal chemistries. One was the cycloalkyne azide cycloaddition, also now often referred to as copper-free click chemistry. But the other was a reaction we called the Pictet-Spengler ligation. And we had developed this chemistry for site-specific modification of recombinant proteins through an intermediate post-translational modification that introduces an aldehyde group. And we called that the aldehyde tag technology. And we can literally genetically encode an aldehyde at the site that we would like this chemical handle to sit. And then it will react with an indole that has this functionality to form a carbon-carbon bonded product, which very stably attaches any cargo of interest to the antibody. And in fact, in the early days, we developed this not for enzyme conjugation, but rather for antibody-drug conjugate synthesis. But it was really useful for this application because the aldehyde tag allowed us to site-specifically introduce a linker with an azide group so that in parallel, we could take the sialidase, do chemistry on it to introduce the cycloalkyne, and then these two functionalities would undergo the copper-free click chemistry to form the triazole product. And it was a really nice way of having like a modular assembly. So we could make antibodies with aldehyde tags. We could take different sialidases and modify them, and then just kind of mix and match them together in a modular fashion. And I think this is really also relevant to this particular seminar series because we formed a company out of Berkeley that licensed the aldehyde tag. And the name of that company was Redwood Bioscience. And we were looking for funding to kick that company off back around the year 2008. And our very first investor was none other than Mission Bay Capital. So they had just launched with their very first modest fund. You know, they had $8.5 million in the bank, and they were looking to seed startup companies coming out of the UC system. And there we were. So we were the very first investment from the QB3 Mission Bay Capital. And that was back in 2010 when we closed that financing round. And then things went very well for us at Redwood. And then in 2014, we were acquired by Catalent Biologics. So now the Redwood antibody drug conjugate technology is a Catalent technology. And this exit for Redwood was the very first exit for Mission Bay Capital as well. So I like to think that the work at Berkeley and the QB3 and the sort of entrepreneurial environment that QB3 fostered really allowed us to translate one of these early technologies from our lab. Anyways, we did the chemistry, we made the conjugate, and then the next question was, can we use these antibody enzyme conjugates to selectively remove sialic acids from cancer cells without having, digestion of sialoglycans off of other cells that don't express the antigen? So what's the on-target, off-target selectivity? And to get at that, Melissa did an experiment where she mixed together in the same tube cells that were HER2 positive and cells that were HER2 negative, but they had roughly the same amount of cell surface sialic acid. And we could quantitate the sialic acids by flow cytometry staining with a lectin called SNA, which binds sialic acids. So this mixed population, when treated with our Trastuzumab-sialidase conjugate, showed us that we could selectively remove the sialic acids from HER2 negative cells without any significant loss of sialic acids from HER2 positive cells. And you can see that down here. Here's the two-color flow plots when the cells are treated with different concentrations of the conjugate. And, at these sub-nanomolar doses, we had significant loss of sialic acids from HER2 positive cells. You can see they drop down from the upper to the lower right-hand quadrant, whereas the HER2 negative cells are pretty much unaffected until you get way out here to triple-digit nanomolar doses. So, our therapeutic window here for on-target, off-target is something in the order of 10 to the 3 or 10 to the 4, which is really great selectivity, we thought. And it allowed us to basically take these conjugates and put them in animal models to see if they could potentiate the immune response against tumors. So here's some data from a model that we ran together with Heinz Laubli's group at University Hospital in Basel. And his student, Michael Stanczak, had basically created a HER2 positive breast cancer model that was a syngeneic orthotopic model in the mouse. So the EMT6 breast cancer cell line is a murine cell line. He stably expressed human HER2 in those cells and then engrafted them in the mammary fat pad. And then Michael would monitor tumor growth as a function of time. And this was a model that was known to be resistant to the naked antibody trastuzumab, and we recapitulated that. You can see the tumors in the black line are growing just as fast as they would if they were treated with trastuzumab in the blue line. So there's no benefit of the naked antibody. However, when he looked at our sialidase trastuzumab conjugates at 2 different doses, there was a pronounced effect, blunting tumor growth. And these little tumors, when harvested, were riddled with immune cells. So they were much hotter than these tumors normally would be in the absence of the sialidase. So data of this type basically gave us the ammunition to start a company around the technology and around the concept of targeted sialidases as an immune therapy. And so I co-founded a company called Palion Pharmaceuticals, which is located out in the Boston area. And the scientists at Palion did some really wonderful improvements on the nature of the molecule. So whereas we had kind of prototyped the system with our chemical conjugate of the bacterial sialidase to the human antibody, at Palion, they re-engineered the molecule. So now a human sialidase called Nu2 replaces the bacterial enzyme. And that's important for a human therapeutic because it hopefully would be less immunogenic than a bacterial enzyme. And also, rather than doing the chemical conjugation as we had done, they figured out how to do this all through genetic fusions, where they basically replaced one of the FAB domains of the antibody with the enzyme and just genetically fused it to the remainder of the FC. So this molecule has a molecular weight that's much more similar to a native antibody, and it expresses well, and it's basically now in the last stages of IND enabling studies, and we plan to put this into humans in 2023. So hopefully this story gives you a sense, first of all, of how bioorthogonal chemistry began, and it was really as an enabling tool for glycoscience, a great example of how an unmet need in biology could drive innovation in chemistry. And since that time, bioorthogonal chemistries have expanded in the options. Lots of people have contributed new reactions and reagents, and they've been applied to many different biological problems and even therapeutic modalities beyond any of the work we did in my lab. So they turned out to be very useful, much more generally. But I think that the combination of glycoscience and bioorthogonal chemistry is particularly powerful because glycoscience is an area where problems don't always get solved with the conventional biological tools, like the molecular biology tools and the CRISPR tools. You need different tools that are suited for glycoscience, and often they come from chemistry. And so, I told you the story of how we invented bioorthogonal reactions and how we use them to make new types of therapeutics, leading to the formation of Palion. But this is just one of several companies that I've either co-founded or that I advise, in which there's a partnership between glycoscience and bioorthogonal chemistry. So, any one of these companies on this list, has a story of its own that I could tell at another time. But I think the point is that it's a really exciting time to be going from basic science to clinical translation. So with that, let me thank the people. I already mentioned former coworkers who did some of the foundational work. I also have plenty of people still in the lab that are kind of pushing the frontiers of cancer immune therapy development and bioorthogonal chemistry applications. But I think the most important thing to say to this audience is how grateful I am to UC Berkeley for taking a chance on a young assistant professor with some crazy ideas about chemistry and living systems, for the brilliant students and postdocs who joined my lab at Berkeley and really did all of the work that is now recognized with the Nobel Prize. So let me end this talk by saying, go Bears, and hopefully we have time for a few questions.
Thanks so much. Thanks so much for a fantastic talk.